All right, welcome to the Explainer. With final exams just around the corner, I know that getting a solid grip on a full molecular cloning workflow can feel, well, a little daunting. So today, we're going to do a deep dive, breaking down every single step of a classic cloning experiment using the pDrive vector, just to make sure you're totally prepared.
Okay, so here's our game plan. We're going to follow the exact sequence you'd use in the lab. First, we'll look at the cloning blueprint. That means really understanding our vector and our insert. Then, we'll head to the virtual lab bench to prepare and assemble the plasmid. After that, it's all about screening our results, confirming we got the right clone, and finally, we'll dig into some of the vector's hidden features that make it such a cool tool.
All right, let's jump right into section 1, the cloning blueprint. You know, just like an architect has to know their materials inside and out, we need to get up close and personal with our two key components, the pDrive vector, which is going to carry our gene, and the PCR insert, which is our gene.
So, our main workhorse here is the pDrive vector. It's a 3.85 kilobase circle of DNA, and think of it as our engineered chassis. It is absolutely packed with useful features. For starters, it has both ampicillin and kanamycin resistance genes. These are our selectable markers, and they're crucial because they ensure only the bacteria that have actually taken up our plasmid will survive. Then there's the multiple cloning site, or MCS. This is basically the cargo bay where our gene is going to go. And the genius part? This MCS is plonked right in the middle of a gene called LacZ alpha peptide, which is the whole secret behind our blue-white screening trick later on. The MCS is also flanked by T7 and sp6 promoters. We need those for making RNA or for sequencing. And finally, it's got a high copy number origin of replication, which basically means our bacteria are going to become little factories, churning out tons of our plasmid.
Okay, but this is where it gets really interesting, the actual map. Check out the table. The T7 promoter, for instance, sits at base pairs 239 to 258, and right after it, the multiple cloning site starts at base 266. What does that tell you? It tells you any gene you pop in there is going to be directly controlled by that T7 promoter. Now look at the LacZ gene. It stretches from 216 all the way to 593, completely swallowing the MCS. This is the whole structural basis for what we call insertional inactivation, and it's what makes blue-white screening possible. Understanding this map with this level of precision is absolutely critical when you're planning your next steps.
Next up, we've got our actual cargo, the insert, our gene of interest. We make millions of copies of it using PCR. But here's the trick. We use custom primers that have these little extra tails on them. These tails contain the recognition sites for two different restriction enzymes, EcoRI and NotI. So as we amplify our gene, we're also adding these sites to either end, like molecular handles, and using two different sites is a really clever move. It forces our insert to go into the vector in one direction only. That's what we call directional cloning, and it's absolutely essential if you want your gene to be expressed correctly. Okay, on to section 2. We've got our parts, we understand the blueprint, now it's time to actually build the thing. This is where we cover the nitty-gritty of preparing our DNA, cutting it up, and sticking it back together to create our brand new recombinant molecule. So this prep phase is super meticulous, and for good reason. It's a 4-step process. 1. Amplify your insert, and you better use a high-fidelity polymerase to avoid mistakes. 2. Purify everything. Get rid of all the junk from the previous reactions. 3. Digest. This is where we use our molecular scissors, EcoRI and NotI, to cut both the vector and the insert, creating those compatible sticky ends. And 4, this step is so important, dephosphorylation. We treat the cut vector with an enzyme called alkaline phosphatase. It chews off the 5' phosphate groups from the vector's ends. Why? Because DNA ligase needs that phosphate to work. So, no phosphate, no religation. This one simple step drastically cuts down on the number of empty plasmids we get at the end. And really, this whole assembly process boils down to these two essential molecular tools. You've got your restriction enzymes like EcoRI and NotI. Just think of them as incredibly precise molecular scissors that only cut at one very specific DNA sequence. And then you have T4 DNA ligase, which is your molecular glue. It comes in and forms those strong phosphodiester bonds, stitching the backbones of the insert and vector together into one seamless recombinant molecule.
Now here's a classic exam question, so listen up. Getting the ratio right in your ligation reaction is everything. We aim for a 3-to-1 molar ratio of insert to vector. And I stress molar ratio because the insert is usually way smaller than the vector. If you just match them by weight, you'd have way too few insert molecules floating around. By putting in a 3-to-1 excess of insert molecules, we're basically tipping the odds in our favor, making it statistically much more likely that a vector will bump into an insert rather than just closing back up on itself.
So, we've built our new plasmid. Great, but right now, it's just sitting in a tiny plastic tube. We need to get it inside a living factory that can make millions of copies for us. That process is called transformation. We take our plasmid mix and add it to special E. coli cells called competence cells, which have leaky membranes. Then, we give them a quick heat shock, which sort of startles them into sucking up the plasmids from the surrounding liquid.
And that brings us to Section 3, screening. So, our transformation worked, and now we have a petri dish covered in bacterial colonies. But here's the thing, the whole process isn't perfect. We've got a mix of cells, some with the right plasmid, some with an empty one, and some with no plasmid at all. Now comes the real challenge, finding our needle in the haystack.
Which leads us to the critical question. You're staring at this plate, right? It's covered in thousands of little dots. How in the world do you find the one, or the few, that actually contain your gene of interest without having to test every single one?
And the answer is just so elegant. It's called blue-white screening. Remember how we said the cloning site is right in the middle of that LacZ gene? Well, if the vector is empty, the LacZ gene works. We grow the bacteria on a plate with a chemical called XGAL. The working enzyme chews up the XGAL and produces a bright blue color. So blue colonies mean failure, an empty vector. But if our gene of interest successfully got inserted, it breaks the LacZ gene. The enzyme doesn't work. XGAL is left alone and the colony stays white. It's that simple. White colonies are the ones we want. Pick the white ones. Okay, so we've picked our promising white colonies. We're feeling good. But in science, we never just trust our eyes. A white colony is a great sign, but it's not absolute proof. So in Section 4, we ask the big question. It's white, but is it right? And we'll cover 3 essential methods to verify it. So we have a 3-pronged attack to be absolutely sure. First, a restriction digest. We grow up one of our white colonies, purify the plasmid DNA, and then cut it again with EcoRI and NotI. If we run that on a gel, we should see 2 bands, a big one for the vector and a smaller one for our insert. Second, we can do a quick diagnostic PCR. We use the plasmid as a template and use primers that we know sit on either side of the cloning site. If we get a PCR product that's the size of our insert, we know something's in there. And third, the gold standard, Sanger sequencing. This literally reads out the A's, T's, C's, and G's of our insert, leaving absolutely no doubt.
And, there isn't one single best method here. It's all about what you need to know. Restriction digests and PCR are awesome for screening lots of colonies quickly. They're fast, they're cheap, and they tell you if an insert of roughly the right size is there. But they don't tell you what that insert is. Sequencing, on the other hand, is a bit slower and costs more, but it is the only method that gives you 100% definitive proof. It confirms the identity, the orientation, and it'll even tell you if any mutations snuck in during the PCR step.
All right, for our last section, let's zoom back out from the nitty-gritty of the workflow and look at some of the other really powerful built-in features of this pDrive vector that you need to be aware of.
So, sitting right upstream of our cloning site is this thing called a T7 promoter. And this is a big deal. It's what we call a strong constitutive promoter. Let's break that down. Strong just means it has a super high affinity for RNA polymerase, so transcription is happening all the time. And constitutive means it's always on. There's no off switch. It just drives expression continuously, assuming the bacterial cell has the right kind of polymerase to recognize it.
But having a promoter that's basically a fire hose stuck on full blast can cause some serious problems, especially if your goal is to make a protein. Pumping out one protein like crazy can actually be toxic to the cell. It puts a massive metabolic burden on it. This can lead to the protein misfolding and clumping up into useless little bricks called inclusion bodies. And, most importantly, you have zero experimental control. The expression is just on. You can't turn it on or off when you want to.
So you might be thinking, well, why would anyone use this vector then? And the key is to remember what it was designed for. Even with that intense promoter, the pDrive vector's core design is absolutely fantastic for the cloning and DNA production part of the job. That high copy number origin gives you a massive yield of DNA. Its small size makes it really easy to get into cells and keeps it stable. It's a workhorse for making lots and lots of your plasmid DNA.
And that really brings us back to the whole point of this. It's easy to get lost in all of these details, the digests, the ligations, the screening, but all these steps are in service of a much bigger idea. The real power here isn't just about making more DNA. It's about isolating one specific piece of the genetic puzzle so that we can finally study it and figure out what it actually does. Which leaves us with one final and maybe the most important question. We did it. We've built and verified our recombinant plasmid. So, now what? How could this one tiny circle of engineered DNA be the key to producing a life-saving medicine, or understanding a complex disease, or answering a fundamental question about how life works? That, right there, is where the real science truly begins.